Hello everyone! The purpose of this video is to explain the expectations for your lab report for Experiment 8, spectrophotometric determination of manganese in steel wool. So I have a Word document here, so you should submit a Word document that looks similar. So I have my title, and you should of course have the name, your name and the date. And the first thing that appears is the abstract. It is the first thing that appears in your lab report. You might want to write it last because it is a summary of the entire report. So sometimes I find it easier to write an abstract last rather than first. In any case, this is what should be in your abstract. You should start out with the objective, and the next thing you should do is you should explain why we used spectroscopy for this particular analysis. We were trying to figure out how much manganese was in steel wool. There's not a lot of manganese in steel wool, as you found out when you did your analysis. So why do we use spectroscopy? Why was that a really good way for us to determine how much manganese was in the steel wool? So the next thing you should do is you should describe the strategy that was used to convert the metallic manganese in the steel wool into the soluble permanganate ion. So there was a whole series of reactions that we did, a whole series of reagents that we used. So you should describe the reagent that we used, and you should describe what that reagent did. That was covered pretty extensively in the pre-lab video, so as resources for this you can use either the lab manual or the pre-lab video. You shouldn't say amounts though. You shouldn't say how many milliliters you added of each of the reagents. So don't include that information, and don't include anything about glassware. So you shouldn't say anything about graduated cylinders or flasks or anything like that. So the next thing that you should do is you should explain how do you go from the concentration of permanganate in your precisely diluted sample to the percentage of manganese in the steel wool. So of course we used Beer's law for that first step. So you should describe the strategy of how you did these calculations. But you shouldn't report any values until the end. The only results that you should report are the percent manganese and the plus or minus the standard deviation. That's all the results you need to report. And then you should conclude with some kind of concluding statement that summarizes the significance of the experiment. And a really good choice, I think, to do this would be revisiting the choice to use spectroscopy as the analytical technique that we used to determine the manganese in steel wool. So that's the abstract. Please confine it to one page. I know there's a lot in this, but be as concise as possible. Abstracts are supposed to be short and concise, but complete. It's a difficult thing sometimes, but that's something you should aim for. The results. Now there are 2 tabs on an Excel spreadsheet that we gave you as a template. So I will go to the Excel spreadsheet. So there are 2 tabs. I'll make this a little bigger so we can see this. So the first tab is labeled standard curve, and this is blank for a reason because you're going to fill it out. So here I have the title and the experiment number and the date. You should put your name right there. So you need to record here the stock solution of the potassium permanganate that we used, your lambda max, and the volume that you diluted the standards to. So down here, you should construct a table. So construct a table like the one you did in your lab notebook that provides the concentrations of the standards. So you should record the volume so that you can then write formulas for calculating the concentrations using the data up here. So you should record volume, do a calculation of the concentration, and the other thing that should be in your table is the absorbance of each of your standards at the lambda max. Don't forget to include water as one of your standards. So you should have 6 standards. The other thing that should be on this tab is a LINEST analysis of your data, with absorbance being the Y data and concentration being the X data. And finally, you should have a beautiful standard curve underneath where you have constructed the graph using all of the best practices that we have been talking about since we had our Excel workshop a few weeks ago. All right, so let's go back to my Word document. Okay, so this is parts 1 and 2. So you can just say on your Word document, please see Excel spreadsheet, worksheet 1, standard curve. And then part 2, please see Excel spreadsheet, worksheet 2, class data. So if I go back to my Excel and I click on class data, what we've provided for you, and I'll make this a little bit bigger, is some overall data. So we all diluted our unknown solutions to 100 milliliters. Since everyone reported their values of the molar absorptivity epsilon, what we did is we went through all of those values and we averaged them. So this is a class average of the molar absorptivity. The path length of our cuvette was the same for everyone, 1.00 centimeters, and that's the molar mass of manganese. So what we have here are all of the samples. So this is just kind of random. If you go through this, we have 34 samples. If you consider everybody in the class, everybody did 2 samples. So you should find your data in here somewhere. I have not formatted this at all. So you should think about how you want to format. See, it says just general up here, and it says just general up here. So think about how you want to format the mass data and the absorbance at the lambda max. So this is net absorbance. So the student values right here are the absorbances. So the absorbances of the blanks have already been subtracted. So you don't have to do that. But what you do need to do is you need to fill out the rest of this table. So you need to use Beer's law to go from absorbance to manganate concentration, then moles of manganate, moles of magnesium, mass of magnesium, and then finally the percent manganese in steel wool. So all of these columns should have formulas. I know there are a lot of samples, but Excel makes this easy. All you need to do is write the correct formulas for sample 1, and then you just need to copy for all the rest. So Excel makes it easier for us. So down here, we have the average and the standard deviation for all of the class data. So you should report those values using the correct amount of significant figures. All right, I'm going to go back to my Word document now. So a lot of what we ask you to do in the calculations interpretations are also in worksheets 1 and 2. But we need to see some sample calculations. So it would make sense for you to use one of your samples for your own data when performing these calculations. And here you can use your own value that you got for your own graph to express epsilon. So we have the slope and the y-intercept of the line, so you'll get that using your line S data. And then you need to calculate epsilon from the slope. So remember, Beer's law is absorbance is equal to epsilon times L times C. So that means that the slope incorporates both epsilon and L. So we need a correct calculation of epsilon using the slope, which has units, and the path length, which also has units. Then the next thing we need to see is the calculation of the percentage of manganese in steel wool. So you'll use the net absorbance of the sample and Beer's law to calculate the molar concentration of the permanganate derived from the steel wool. From there, you need to show us the total number of moles of permanganate obtained from the steel wool. From there, use molar relationships to determine the number of moles of manganese from the permanganate found in the original sample of the steel wool, and then convert moles to grams, and then you can compare the grams of the manganese to the mass of the original steel wool sample, multiply by 100 to get the average, get to the percentage of manganese, and then you could report an average mass of both your samples if you'd like to do that. Remember, for all of your sample calculations, you have to you present an equation, an explanation, something that orients the reader to what you are calculating. You don't just dive in with the numbers. So give us the equation, spell everything out, and then do the substitution. Make sure that your units are there. Make sure that your significant figures are correct. This is a full report, and full reports are supposed to have questions, and we did notice that there were no questions. So it would be interesting for you just to answer this one question. So examine the class data and write a short paragraph in which you account for the range of results in the data. So we all use the same sample of steel wool. We all should have gotten exactly the same thing, and we didn't. So there must be a reason for that. There must be some way that you can use your knowledge of chemistry to account for the range of the results that we got. And of course, you would end with references, so don't forget that. So what you'll do is you'll submit to the our Sakai site this Word document that you'll have completed, and this Excel document where you've completed both this tab for the standard curve, again, so all of the data for your preparations of standards, the line nest, the standard curve, and the completed worksheet for all of the class data. So I hope this is helpful, and if you have any questions, we'll see you in office hours.